This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 279, recorded on December 29th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello. And after the bitter cold winds blew through, today it is 50 degrees Fahrenheit.
Isn't that amazing? It was so frigid.
My goodness. But now it's unseasonably and kind of freakishly warm.
Yes, I'm happy with that. Also joining us from St. Louis, Petra Levin.
Hello, it's good to be here. I think we've had a 65-degree temperature change in the past week, and all our snow is gone.
Oh, you had snow. Did you have snow in Ann Arbor, too?
Yeah, but only maybe 2 inches.
Yeah, that's like in St. Louis, that's almost enough to shut everything down.
Yeah. We didn't have any snow, but we had that cold weather where you go out to walk the dogs and they come right back in.
Right.
Because usually they want to fool around outside and chase small critters, but nope, it's too cold. So we have to go out with the dogs because they're small, and we have a lot of hawks. We're afraid the hawks will pick up the dogs.
Oh, scoop them. Yeah.
That's happened. That's happened in our town. Can you imagine seeing your dog being taken up into the sky? That must be terrible.
I have seen a lot of hawks with squirrels, but my small dog is on the end of a leash, and my big dog weighs 60 pounds. So
the big one's no problem. Yeah. What kind of dog is the small one?
She is a mini schnauzer, and she has a very strong opinion about small animals. She does not like them. So
terriers are bred to chase small animals.
She sits in the kitchen and cries at them.
We did a paper on TWiM last week where they looked at the genome sequences of many different breeds of dogs, right, and tried to correlate the genome with the different properties of the breeds, and the terriers chasing small animals was one of them, and herder dogs that would herd sheep and so forth. They could see distinct signatures from these different breeds. Very interesting paper.
Well, the really Schnauzer is really interesting because it's the only, I think, non-English terrier. So it was bred completely independently and yet came up with the same characteristics. So that would be a good outgroup, I think, for comparison.
I don't know if they did that in that paper. I don't remember, but it was a lot of fun. But we are not here to talk about dogs today. We are here to talk about microbes, and we will. This is the last episode of 2022. Probably did maybe usually 48-ish episodes, 2 a month. No, that'd be 24. Roughly 24. It was a good year, and I hope you enjoyed your microbiology this year, and stay tuned next year. There'll be more. But I'm going to start us off with a snippet on a subject that I really enjoy. I've done a number of these papers on phage therapy for infections on this podcast and TWiM. We actually had a fellow at Yale whose name is escaping me who does this as well on our podcast. But this one was published in Cell. It's called Host and Pathogen Response to Bacteriophage Engineered Against Mycobacterium Abscessus Lung Infection. First author is Jerry Nick, who is also a corresponding author along with Graham Hatfull. Comes from a number of institutions: National Jewish Health in Denver, University of Colorado, University of Pittsburgh, the Colorado State. And, as many of you may know, Graham Hatfull has developed a program where high school students collect dirt and they isolate phages of mycobacteria, not abscessus, but I think they use smegmatum. And he uses some of these phages for these experiments, as you'll see in this paper. And so this paper is all about treating a Mycobacterium abscessus infection. This is a non-tuberculous mycobacteria, which can infect people who have a number of issues. And as you will see, people with cystic fibrosis are particularly vulnerable to infection with non-tuberculous mycobacteria. And these, they get a number of infections, of course, but when they have these non-tuberculous mycobacteria, NTMs, this makes them poor candidates for lung transplants, which is one way you can solve the problems of patients with cystic fibrosis. And that is because they often have other infections, and so antimicrobial treatments doesn't work well because of the NTMs. The mycobacteria can survive in microenvironments throughout the lungs, very difficult to treat. And so in this paper, they explore phage treatment of M. abscessus in a patient with cystic fibrosis. Now, some of the problems include that every patient's isolate is slightly different and has different susceptibility to phages. So you have to tailor the phages to the isolate of the patient. Many of these phages are temperate. They don't lyse the host, the bacterial host. So you have to turn them into lytic phages, which can be done by engineering, by modifying the genome. And so this is what they do in this paper. They have a young person, a 26-year-old man with cystic fibrosis. They're going to treat him with 2 engineered mycobacteriophages. And his infection was refractory to multi-drug antimicrobial treatment. He was considered not a candidate for lung transplant. They treated him with this 2-phage cocktail, and voila, he became a candidate. He got a lung transplant as a consequence of eliminating this M. abscessus with the phages, which is really remarkable.
And the reason for that is when they do any kind of organ transplant, you have to go on immunosuppressive therapy, so your immune system gets dampened. So if you're harboring a few refractory microbes, when you're on that immunosuppressive therapy, you could then have a relapse and a poor lung infection.
Yeah, absolutely right. That's exactly it. And so this is an experiment to see if we can treat this M. abscessus infection with phages. So as I said, 26-year-old man with cystic fibrosis. He also was infected with multi-drug resistant Pseudomonas aeruginosa and methicillin-resistant Staph aureus.
Wow.
Yeah, no, that's those ones.
I mean, this patient is in and out of the hospital for years and years and years. This story begins in May 2016. He first turned positive for M. abscessus, and that was 1,500 days before initiation of phage treatment, so many years before he even saw a phage. Over the next 4.7 years, he was maintained on 4 to 5 drugs, rotated based on the availability of the drug, was in and out of the hospital. But he had 11 hospital admissions ranging from 11 to 21 days in duration, really no microbiological response to this treatment. And he was declined for lung transplant by 3 independent programs due to no way of treating the M. abscessus.
And I believe those 11 hospital stays were over a 1-year period.
Okay.
So 2 to 3 weeks a month in and month out.
Yes. I mean, just consider what kind of life that is, right, to be in and out of the hospital so frequently.
Yeah.
It's really, I cannot imagine someone's mental state who has to do that, right?
Right. So clearly we need new therapies, better therapies.
For sure. So they got an isolate of M. abscessus early, 1,500 days before phage administration. They did the genome sequence to see what they were dealing with. They screened it for sensitivity against a panel of phages and found phages that would infect it. And they used, of course, lytic derivatives. So these the lysogenic phages had been engineered to become lytic, and they found 2 in the end that were chosen to be a mixture, a therapeutic phage cocktail. They grew them up, they purified them, showed they were sterile, showed they were endotoxin free, and in September of 2020 began to give these to the patient. 10 to the 9th plaque forming units of one, 10 to the 8th PFU of the other, given intravenously twice daily, and continues to this day, by the way. As of March 2022, when they wrote the paper, they said, we're still doing this. 5 days after initiation of phage therapy, he was discharged from the hospital, then readmitted 4 times in the next year when he had various exacerbations. But a number of cultures, sputum cultures and so forth, indicated that his infection was controlled. The M. abscessus infection was controlled after this time, and he then was listed for a transplant and got his transplant on day 379 after initiating the phage therapy.
Which is right there. That is a huge success. Yes.
Amazing, right?
Yep.
So nothing else worked. And his Pseudomonas and his MRSA were not controlled by the phage. They looked at that. This was, and it made him have a lung transplant. So yeah, it's a success by that, because otherwise he probably would have died.
Right, and I think the Pseudomonas and the Staphylococcus are probably part of the CF.
Yeah.
And so when he has the lung transplant, I think most of the time that problem is
those. Yeah. So of course, when you remove the lungs, you can then look in them for infection. So they did a variety of experiments on the lungs to look for evidence of M. abscessus. They had swabs, they had growth, they had pieces of the lungs, they had pathology that they could look at. And so they stained for what we call acid-fast bacilli, of which M. abscessus is one. And all the samples were negative in all the lobes of the lungs that they looked at. They didn't see any pathology consistent with acid-fast bacilli. There's a typo here. It says acid-fact bacilli. It should be acid-fast. And they didn't see any evidence of infection. They say it seems to be no detectable M. abscessus in these lungs. They also did PCR, and the PCRs of samples later in the treatment. So they have all of his sputum samples throughout the treatment. Very low levels of material below the limit of detection, which they say is consistent with primarily negative cultures in that same time frame. So they also had done cultures which seemed to be negative, and the PCR is very, very low. So probably just pieces of DNA here and there. They also looked for antibody titers to M. abscessus. I'm sorry, they looked for antibodies against the phage and the M. abscessus. Sorry, it's both of them. Both antibodies against the phage and the M. abscessus decline over the course of infection. And they find no phages in the bloodstream. They find no phage DNA in the sputum samples. They also did whole genome sequencing on the number of M. abscessus isolates over the course of the infection. And they look at the extent of genetic change, and they find that there's a decrease in what we call accessory genes that are variable and can change from time to time. And they see a decrease in accessory genes over time, which they say indicates a reduced genomic diversity and a selection for a population that's more homogeneous as a result of the phage treatments.
That was important because there's a theoretical possibility that you could apply a selective pressure like the phage, and then you begin to select variants that could be problematic. But they found the opposite.
Right. Yes,
which is really hopeful. Yeah.
They looked at antimicrobial resistance of M. abscessus isolated from the patient. So they tested a 20-drug panel of antibiotics that had been used against to treat the patient, and found no evidence for greater antimicrobial resistance. And they also looked for phage-resistant isolates, right? Because one of the concerns you have is that extended treatment with phage may select for bacteria that are mycobacteria that are resistant, but they don't see this even 8 months after treatment. And another issue that has come up previously is, if you give someone phages intravenously for so long, they're going to make antibodies against the phage. And could these be problematic? It's previously been shown that phage neutralizing antibodies do compromise phage therapy. Interestingly, in the serum from this patient, there was already some cross-reactivity to the phages that were used for treatment before treatment, right? So this person had encountered phages, which is not surprising, right? Because we have a microbiome with their own phages.
Well, I mean, they have an M. abscessus infection. I'm guessing some of those might be lysogenic for some of these phages.
It could be, yeah. So this person had encountered phages that induced immunological cross-reactivity, but they didn't hamper the treatment. And in fact, even with the phage treatment, it didn't increase the antibody titer until later in treatment. So in this case, it wasn't an issue, although it's been an issue before. And I mean, this is something we have to look at, have more experience. There are only a handful of experiences, so the issue of phage neutralizing antibodies needs to be addressed. And so that's the story here. It's a great snippet. Looks like the phage therapy, as they say, viable M. abscessus was sterilized from the lung. Very strong use of words, right?
Mm.
And made this person eligible for lung transplant. No resistance to phage, no alteration in antimicrobial sensitivity. And they think that using 2 phages was important here because there was there were previous patients treated for Pseudomonas and Acinetobacter. They developed resistance to the phage, but they only used 1. And so here they used 2, and they think this may be the way to go. It's kind of like multi-antiviral therapy has been the key for HIV and now hepatitis C virus as well. So they say this could be a roadmap, what we have done here for treating advanced cystic fibrosis and getting patients to the point where they can have lung transplants. So I think it's really exciting.
It's very cool. And it's actually, this paper is the same group or parts of some of the same authors were on a paper from 2019, which is in Nature Medicine, where they used them to treat a patient who had disseminated drug-resistant M. abscessus after their transplant.
Right.
And they couldn't get rid of it because they're immune suppressed also. And they used phage therapy to treat that. So it's a sort of the flip side, I guess, of this in a way.
Yeah,
but what was really, I used this paper with my undergrads because those phage that they used in this paper from 2019 by Diedrich et al., and it's cited actually in the paper you just did as a snippet, those phage were isolated as part of this phage hunters or C phages course that Graham Hatfill actually kind of organized, and that WashU uses. A lot of schools use this for their undergrads, and some of my students have taken this class, and they had isolated these as smegmatis phage from the soil. And these phage have names. I think the one that worked in the end is named Zoe because the students name them. And so I thought this was a really beautiful follow-up to that 2019
very cool.
paper.
Yeah, we
so
did that.
They're biobanking these phage, the C phage. Yeah. Students from all over the country.
Can you imagine how a high school student learns that their phage has been used to save someone's life? How cool
amazing.
It must be. Right?
Yeah, it must be very cool. And I think the original one was even students from, I think they might have even been from Australia and not from the US. So again, C phages is all over the world. And since they've been collecting smegmatis phage, they can use them they work against also abscessus.
That paper you're mentioning, we did that on, I think, TWiM. The cool thing was halfway through, they had to re-engineer the phage to match the isolates, and they did that. So you can, this is such a cool thing that you can change it to suit.
It's very cool.
By the way, the guy at Yale is Paul Turner, who's also been involved with treating patients with phages. So
the other thing that struck me about this paper is just the care in the design. It's clear that they're showing utmost respect for this person, the patient that they're treating, and they're being as thorough as can be. They're not using just one assay, for example, to follow the colonization by the bacteria, but they're using, multiple independent quantitative tests. They're biobanking all these samples. So they're doing everything they can to really learn as much as possible from this one patient so that we can then use that to guide therapy.
It's one patient, and that's important to point out. It's one patient, and so what happened could be different in the next patient because everybody's different, right? So
and the microbes are different, right? And who else is colonizing is different, and the interactions may change things. But what a contribution they've made, not only to our basic understanding of these complicated infections, but also to treatment and potential applications for therapy.
Yeah, it's very exciting. I think obviously more work is needed, but it looks promising, right?
Strike one up for the phages, Vincent.
That's right. People say our virus is always bad, and here's a great example of when they can be used for the good.
Yeah.
All right. Petra, you have a paper for us.
I do have a paper today. So this paper is entitled Encapsulation of the Septal Cell Wall Protects Streptococcus pneumoniae from its Major Peptidoglycan Hydrolase and Host Defenses. It's by Joana Figueiredo, Malfada Xavier Enriquez, Maria Joao Catalao, Sarah Pinheiro, Ana Rita Narciso, Francisco Mesquita, Bruno Manuel Saraiva, Madalena Carrito, Didier Cabanas, Mariana Gomez Pinho, and Sergio Raposo Felipe from Portugal. And I think the title aside, this paper actually addresses some really important questions that are, I think, broader and worth thinking about generally. So capsule is a polysaccharide on the surface of many bacteria. It's known to be a major virulence factor for pathogens. So it's on the surface, so it can protect them from the immune system, but it's also a target of the host immune system. Because of this, some bacteria have many variants of the capsule. It's made of different sugars, and they can modify them in different ways. So there are these polymerized sugar chains. And so if a host becomes immune to one serotype of variant of capsule, it won't be immune to other variants or serotypes. And Streptococcus pneumoniae has hundreds of capsule variants. There are hundreds of serotypes of this organism. So Streptococcus pneumoniae, you might remember if you've taken intro bio, because this is the organism that was used in a 1944 paper by Avery, McLeod, and McCarty, where they demonstrate that DNA is the likely molecule of heredity. They do this. They have two variants of S. pneumoniae. One is called rough because the colonies are kind of dry and rough looking, and one is called smooth because the colonies that the kind of wild type virulent one form have the kind of shiny surface that's actually from capsule. And actually capsule is very sticky. If you kind of streak these guys out, I think the best description is they're kind of snotty. And that's just this sugar capsule on the outside of each of the bacteria. And only the smooth ones in this paper, which if you haven't read it, it's actually a beautiful paper to read. They're very thorough, and they kind of don't believe their own results, which make it even more wonderful. But the smooth variant actually can kill mice, and the rough variant can't. And they show that basically if they break, so S. pneumoniae is what's called competent. It can take up and integrate DNA. And that's sort of an advantage for this experiment, because they basically take up the smooth bacteria and they grind them up and divide them into their constituent parts, into capsule, protein, DNA, lipids, and they mix them with the rough ones when they infect the mice. And they find only the DNA actually has transformation. It can make the rough ones smooth so that they can actually kill the mice and make them sick. And again, that's because of the capsule. And what's really, I think there are a couple of interesting things about capsule. One is, although it's important for virulence, important for pathogenesis, both as a way to evade the immune system, but also a target for the immune system, bacteria that never see a host also have capsule. So one of the questions is, why have capsule if you're not using it in a host? We sometimes get very focused on pathogens, but it's true that all bacteria probably that have capsule, that's not why it was invented, let's say.
Well, so one of the ways that the capsule protects the bacteria in our bodies is it makes it much more difficult for white blood cells to grab on and ingest the microbes and kill them. And out in nature, there are lots of amoeba and protozoa that are also ingesting and digesting bacteria for food. So one simple selective pressure would be environmental microbes are also encapsulated to protect them from being ingested by predator amoeba.
Right, that would make perfect sense. That's definitely another function. And I think it's good to think about it more broadly that way, because they'll also get to another reason why capsule might be protected kind of at the end as almost like an add-on. But this paper really addresses a couple big questions about capsule. One is capsule is like an outer layer of the bacterial cell envelope. So in gram-positive bacteria, which S. pneumoniae is, there's an inner, the plasma membrane, like all cells have the cell membrane. Then there's a very thick cell wall that's made of a molecule, giant molecule called peptidoglycan. And then there's capsule, and capsule is actually covalently linked to that peptidoglycan in these organisms. So in gram-negative organisms like E. coli or Salmonella, capsule would be linked to the outer membrane. But in gram-positive, it's linked directly to the cell wall through a covalent linkage. And all of these layers are very uniform. And that means that they must be regulated together and coordinately so that you have this beautiful sort of layered structure where the capsule is even thickness and all the way around the cell, just like the cell wall. And so this paper addresses kind of two big questions. One is, how is the capsule or machinery localized? How does it get to the cell wall? And specifically in this case, the cell division site. And two, how is synthesis regulated? How is it coordinated with cell division and getting to the septal site? So they're really, this paper is a little hard to read only because the gene names are all very similar. In this case, there are three players that you really need to keep track of here. WZE, which is a regulatory protein, which can autophosphorylate. It can put a phosphate on itself. And when it does put a phosphate on itself, that protein, WZE, can activate capsule synthesis at the septum, where the cell is dividing, presumably by capturing a machinery for capsule synthesis, which includes something called a flipase, which flips individual subunits out, a protein that polymerizes and links the subunits into these long carbohydrate polymers that make the capsule, and then a protein that anchors the capsule to the cell wall. And again, this is very important to keep capsule attached. You have to actually covalently link it to the cell wall. And this ligase is called WZG. So there's E, which is an activator, G, which actually does the ligation of capsule and attaches it to the cell wall. And there's also a phosphatase, which you don't need to remember, but actually can take the phosphate off E and turn it off. So it can turn it on and off. The third protein, so there's E, the activator, the ligase, G, and then a protein called WZD, which actually co-localizes with E. So D localizes with E at the division site, and it actually activates the autophosphorylation. So E won't autophosphorylate itself. I mean, it will, but it needs to kind of get a little touch or poke by D in order to do it. So before this paper, it was known that D and E localize to the division site together, and they're dependent on one another. And how these other proteins, which have to get to the division site because that's where cell wall is being synthesized, get there and activated was unclear before this paper. And so,
and that paper was also done by this group, also led by
it's a 2011, I think, paper. Mm-hmm. We can put it on the site if people would like. So they start going very broad. So they do something called a bacterial to hybrid to identify proteins that interact with WZD. And this 2 hybrid, very briefly, like most 2 hybrid assays, is basically a way of having, you can put a protein into a cell, in this case you put it into E. coli, and then you can put potential interacting proteins in there as well. And fused to the protein that's your target, that you're interested in finding interactors for, which you call bait, is in this case, half of an adenylate cyclase molecule, a protein that can make cyclic AMP. And fused to all the potential fish, the proteins that you're looking to interact with your bait protein, in this case D, is the other half of this adenylate cyclase enzyme. And so when D finds fish, when the bait finds something to interact with, you bring together the 2 halves of adenylate cyclase, and this makes cyclic AMP. And when cyclic AMP is there, the E. coli strain in which they do this can essentially ferment sugars from lactose to maltose. And in their assay, this leads to the production of beta-galactosidase, which they can measure. And figure 1 is actually this assay where they show the results of this. And so it's pretty simple to read. So when they find an interacting protein in figure 1, they can see that they're interacting together because they get production of beta-galactosidase. So they have a control here on the left where they're putting things they know interact here. And then they have a negative control is the second one in that lane. And then you can see these different tests down at the bottom. And the 18 and the 25 are just the different parts of the cyclic.
And it's a terrific assay because it's cheap and quick, but also their dynamic range is over like 50,000 units. A lot of sensitivity.
This is a great version of Bac2 hybrids. Sometimes they're not quite as clear.
So it allows you to be unbiased and just ask, what proteins in the cell are interacting. It's work, but you can screen efficiently.
Right.
And get quantitative data.
They do a great job here. And they find actually that they only find one new protein that interacts with WZD in theirs, and that's the ligase WZG. So as they would have predicted, at least in this assay, WZD, the protein that activates capsular synthesis at the septum, interacts directly with the ligase, the thing that attaches the capsule to the cell wall.
Neat design.
Yeah, it is very nice. And also it's beautiful that that's what they got because then they have a whole bunch of questions. First, because WZD is known to go to the division site and they speculate that's where it activates capsule synthesis, their first question is whether in the cell, sort of in reality, not just in this Bac2 hybrid in a totally different bacterium, but in reality in the cell, is WZD capturing this ligase and bringing it to the right place for capsular synthesis? And they do this using a fusion, in this case, of a GFP, so green fluorescent protein, so they can see it's localized in live cells. That's actually designed to work well in S. pneumoniae. And they fuse this GFP to WZG because they want to know, is WZD required to bring G to the septum? And this experiment actually, I think, is really easy to see, and the data are actually pretty clear. They're actually measuring relative difference, cytoplasmic GFP to GFP on the periphery of the cell, where the cell envelope is and where the division septum is. And so I think they have actually really nice violin plots for quantification in figure 2. And you can see that when they have wild type cells, the GFP fused to WZG to the ligase actually very nicely gets to the division septum. If you kind of zoom in, you can see these clear bands where the cells are dividing of the WZG. And that's reflected in the quantification.
And because what's happening there biologically is that's where the new peptidoglycan is being inserted into the growing cell.
Right. And so new peptidoglycan, right, you need to cover that peptidoglycan. It needs to interact with and be ligated to capsule. So actually there are two machineries that make peptidoglycan in cells. In these cells, there's some that help make it longitudinal, like on the wall that grows outwards. And then there's another peptidoglycan machinery that makes the cross wall. And all of that has to be covered with capsule for this thing to work. And so this is looking at one of those machineries and its interaction with the ligase. And again, the quantification is really nice, and you can see very clearly only when everything is there do you get really good localization in figure 2. Also, these experiments were done really beautifully because often when we do complementation, we have a mutation in the chromosome and we put one thing on the plasmid. But here they actually put the genes on the same plasmid so they're co-expressed, and they can look for both necessary and sufficient doing this. So they can make sure that that WZG is not only necessary, that requires WZD for localization, but that WZD is sufficient. Okay. All of this is really great. They show that the Bac2 hybrid actually did tell them something that's physiologically relevant inside the cell. You also need these things. Their next question is to look for mutants. that might interfere, because now they're trying to understand mechanism. What parts of WZD are actually interacting with this ligase? And for that, we look at figure 3. And in figure 3, they have a really nice kind of schematic where they've labeled the different parts. There's the N terminus, the beginning of the protein on the left, and the C terminus on the right. They have the different parts of the WZD protein, and they have this little kind of loop that goes from the inside of the cell, outside, and then back inside the cell. And it turns out that in work from a different lab, previous work, that people had identified mutations in that domain, this little loop domain of WZG and WZD. So they identified this to look for interactions, and people had found specific mutations in WZD that didn't interact with, that had problems with getting capsule to ligate. So again, WZG is required for ligation. So it follows that in order for WZG to get to the right place and do ligation, it's going to have to interact with WZD. So when you don't have WZG, you make capsule, but it's not stuck on the cells properly. So if you have mutations in WZD that don't bring WZG to the right place, then you're not going to get ligation of capsule at that place, which is the septum. So here they first do back to hybrid to look for interactions between WZG and itself, as well as these mutations in WZD that interfere with capsule ligation. And what they find is that one residue in particular, residue 56, it changed from WZD valine at that position to an alanine at that position, interferes with their interaction in the Bac2 hybrid. So they can, now you've got, not only is it just the whole proteins interacting, just one change in WZD is enough to interfere with interaction of WZG, the ligase. And this is really cool because, again, they pick these mutants based on their phenotype, the lack of ligation or full ligation of capsule in that mutant. And then it turns out that that's a perfect pick. So they had some hints. And I think actually in figure 4 that they do some localization showing that the mutants don't localize WZG properly. First, the WZD mutants they need to screen. So there's a picture of this little loop in WZD, and they look for localization of WZD in this figure 4. And what they're looking for is to see if the mutations that are interfering with WZD localization are interfering, or are they interfering with WZD's attraction for WZG? So again, you can have WZD, the activator, localized but not interact with its target, the ligase. Or you could have WZD, the activator, not localized at all. And so in figure 4, they're trying to figure out which are the interesting residues, the ones that might interfere with interaction with WZG, but don't actually interfere with WZD's localization. So they maintain WZD's ability to get to the septum, but they interfere with its ability to recruit the ligase. And
so the one you originally mentioned, the valine to alanine, that's 116, right?
The valine to alanine, these are all it's 56A, because 116 you lose septal localization of WZD.
Yeah. Okay.
But if you look at the next one to the right, so that's in the middle of this figure, WZDV56A, that one localizes beautifully, even though it doesn't interact with WZG.
Got it. Yeah. So the other one, 116, is just diffuse. It's doesn't go anywhere in particular, right?
It doesn't go anywhere in particular. You can actually, if you zoom in, you can see that it's very diffuse around the whole cell periphery because it's still a membrane protein, but it's not concentrated at the division septum.
Nice.
And they also have another protein, WZD8Y82F, which also goes to the septum very nicely and looks like 56A.
Now, Y39C is no longer membrane bound, right? Because it's in the transmembrane segment. Is that correct?
It's actually, turns out they say in the text that it's degraded.
Degraded. Okay. Yeah. Because there's no signal, right?
Yeah. Which makes sense. There's no signal because it should be still in the membrane because it's got numerous membrane loops. So when you don't see signal, it's probably degraded.
Got it.
Yeah. Okay. So they have point mutations that don't interfere with localization of the activator, but do interfere with its ability to interact with its target, the ligase.
Mm-hmm.
And that
actually should be doing the job of sticking the capsule at that septum site.
Exactly. So they can get the activator there, but it doesn't mean anything if they can't get the ligase there. Mm-hmm. And so figure 5 kind of really reconfirms this because now they're looking, they did the Bac2 hybrid, and they do the localization again, and they look for localization of WZG in the presence of wild type and mutant versions of WZD. And they specifically use 56A and 82F. And so those are the mutants we talked about where WZD still localizes. And what they see is that in the absence, in the first one panel, if you look at the fluorescence microscopy, the GFP labeled WZG in the absence of WZD is just peripheral. So it's not going where it's supposed to go. If you go one over to the right, where they complement it with wild type activator WZD, now they see the localization they expect, where it's concentrated at the septum. In the third one over, they have V56A. So again, this is the WZD mutant that still localizes properly, but did not interact properly with WZG in the Bac2 hybrid. And when they look at WZG localization in there, it looks essentially like it looks in the WZD null. So in terms of the ability of this ligase, this capsular ligase, to get to the septum, it's essentially looks like it can't get there at all because of this one mutation in 56A. And also, I think interesting is when they make that Y82F mutation, they still see YZG, the ligase, getting to the septum. So that mutation, point mutation, seems to still be able to recruit WZG, but potentially is defective in its activation.
They're learning a lot from these really elegant essays, aren't they?
Yeah, they're really getting the most out of these assays, and I really love that they leveraged this previous paper where somebody had identified mutations that lost the ligase ability or partially lost the ligase ability, because that allowed them to look at only 4 mutations instead of having to scan every residue in that loop, which would have taken a very long time.
You could save yourself weeks at the bench by a couple hours in the library.
Exactly. The classic weeks at the bench. Exactly.
Yep.
Read the literature. Maybe somebody did it or can help you. So these are really beautiful experiments. They totally establish that the way, in this case, pneumococcus or strep pneumoniae has been able to get capsule to the right place and lock it in is through this little activator, which knows to get to the septum. So you have one thing that gets to the septum, and that can bring what else you need to the septum. So it works super well. And at
this point, you don't know whether it's just cell economy, like you don't want to waste those extra capsular polysaccharides and have them not attached at the right place, or is it critical that they mask the new peptidoglycan?
Right. So you don't know why they need to do this, because if you have some capsule, why isn't that enough? And then the other question I think that's actually pretty interesting is why not just have it interact with peptidoglycan synthesis everywhere? Because there are these different machineries, so it has to regulate it in both these machineries, because there are 2 sets of machines that build peptidoglycan, and you need to get capsule to both. So I like that also from a cell wall synthesis kind of side. Okay, but as Michelle noted, the big question in biology is why. This is a lot of elaborate stuff to make essentially a capsule, like a coating on the cell wall. And so they want to know, first of all, is this changing where capsule goes? Because you could imagine that, there's enough capsule, it's kind of going to smear all over everything. And so they localize capsule in the different backgrounds. And they do this using a protein, a peptidoglycan hydrolase, something that cuts cell wall, called LIT-A, which is actually there are a lot of peptidoglycan hydrolases. LIT-A is pretty common. They use one that comes from pneumococcus or pneumoniae that already makes it. And they speculate actually that the capsule is protecting the cell from maybe these enzymes, which are secreted in some cases not secreted, but in some cases secreted by bacteria to essentially destroy the cell wall of other bacteria. So to test that, they use S. pneumoniae's own version of LIT-A, soluble form, and they attach it to GFP. And GFP, the LIT-A's job is to find cell wall, it'll bind to it. And so by doing this, they can look and see, can LIT-A find exposed cell wall, and does it find it differently in the absence of capsule? And I would not be telling you this except for it works super well. They can actually see LIT-A localizing in the capsular mutants. And this is in figure 7. And this is really amazing because, again, this is an enzyme that can destroy the cell wall. And when they localize it, you can see in the wild type cells in figure 7, panel A, it doesn't really light up anything. There's maybe a faint little bit, possibly at the septa. It's really hard to kind of match things up. In the mutant missing WZD, again, the thing that makes sure that capsule synthesis happens at the septum, you can see that LIT-A, this GFP, can kind of find its way into the septal area. And when they delete capsule altogether, the LIT-A is it's like a light bulb, super bright. The LIT-A GFP is just all over the cell. So I think this really says that the capsule can protect from this semi-artificial situation, but it's not actually that outlandish because we know, again, that bacteria make these hydrolases and secrete them to kill their enemies or competitors. They do one last experiment. So one other question is, maybe in the host, though, this doesn't matter, especially if the host doesn't have other bacteria infecting. So, and they show that actually the absence of capsule synthesis at mid-cell does impair virulence. They use zebrafish embryos, and they infect them with encapsulated, unencapsulated mutants and a WZD null mutant of S. pneumoniae. And 30 hours post-infection, essentially all of the ones that were infected with the wild type are dead. A third of the ones that are infected with the WZD null mutant strain, though, can survive. So just having that small amount of exposed surface is sufficient in this zebrafish embryo model for the embryos to essentially kill the bacteria before they themselves are killed.
And that's because the host cells have evolved mechanisms to detect minute quantities of these microbial patterns, including peptidoglycan. Mm-hmm. So there are NOD-like receptors, toll-like receptors that can detect microbial products and then trigger an inflammatory response to clear the infection. So if you're a bug that needs to colonize a host, you need to not expose your peptidoglycan. And what better way than to link your capsule attachment enzymes to the site where peptidoglycan is being added.
So why isn't capsule detectable by the host then?
I believe because polysaccharide is similar enough to the sugar polymers that coat our tissues and create organs that they have not evolved as a ligand for toll-like receptors.
But there must be some cost because not every bacteria puts a capsule up, right?
Right. It's expensive to make, but also we know that as Petra pointed out, we can raise antibodies against polysaccharide. Once you get to be a toddler, 3-year-old, you can raise antibody, and then it's antibodies that decorate the capsule and mediate clearance of encapsulated organisms.
Right. And I think what's important about not having capsule is that your immune system that has a fast response.
Yes.
And responds generically to all gram-positive bacteria in this case can see it. But if you have capsule, you're essentially relying on the host to raise a more specific immune response. And so that takes time. Actually, the figure 6 actually looks kind of where capsule is made in the different mutants. And so you can see, and this is using antibody, I believe that was against the capsule specific in this strain. And you can see that the capsule is actually differentially distributed in the different mutant backgrounds. And in fact, 56A, it looks like capsule is just stuck. So even though it can't get the ligase there, it looks like there's something that reacts with the antibody that actually is kind of stuck at the division septum. And that also is kind of cool because it looks like somehow WZD is recruiting other components, but it can't kind of get it to get out and get into the right place.
So they're beginning to define this really sophisticated, elegant machine that coordinates cell growth with decoration of their cell wall.
Yeah, exactly. Capsule. And they've identified this one interaction with the ligase, but looking at this figure 6, it looks like there's some other things that have to happen, like maybe the ligase needs to get there, and by the ligase getting there, that might help activate the flip base that puts it outside and the polymerase that polymerizes it, because it looks like somehow maybe it's not through a direct interaction with WZD, but maybe with its other ZE or some other interacting proteins, things are kind of going to the right place, but they're just not being displayed on the surface. And I think it's almost like it's sucking up all the capsule to that division site in that figure in the panel A, second from the bottom.
I was also intrigued that in their introduction to this paper, they point out that identification of a particular bacterial protein that is required for protecting and hiding the bug from the innate immune system could be an awesome target for a new therapy.
Mm-hmm. Yeah.
So they don't develop that idea in the discussion, but they certainly raise that as another motivation for these studies.
Right. If you can target capsule synthesis, then the innate immune system can essentially be more effective.
Or targeting of capsule to the growing peptidoglycan makes the cell vulnerable to clearance by the innate immune system.
Mm-hmm. Yes.
So
you're saying devise ways to prevent the shrouding of the bacterium by capsule, right?
Yeah, to mimic that one amino acid change.
Yeah.
Or potentially, I wonder if you could get antibodies to WZD and essentially protect it.
What about an inhibitor of the ligase? You could probably get that, a small molecule, right?
Yeah, I'm guessing you can, although I don't know how that ligase, one question would be, because as Michelle pointed out, these kind of sugar polymers are very common. Yeah. I'm wondering, and I don't remember what the linkage is to, but it could be that there's something that does this similar.
Host.
Yeah, in the host.
Yeah, sure.
Yeah.
You would find that out quickly, I guess.
Yes.
But the ligase that attaches the capsule to the peptidoglycan, you might expect that to have more specificity for the microbe.
Maybe.
Yeah. I don't know, remember what it was linking to.
Yeah. Mm-hmm. Well, this ligase has been known for a while, right?
Yeah.
So it's out there if people wanted to make it a target. So maybe it is too similar to a host ligase or not. I just don't know.
I really appreciated their schematic representation of their design of each molecule that they were going to study, and then demonstrating quite a lot of quantitative data in a way that we could easily absorb it.
Right. I do think this is really beautiful display of data that it's difficult to display localization data, and the numbers used to be sufficient to put in one or two pictures and call them representative. But for the good of, I think, the field, the subcellular localization data in bacteria has become extremely quantitative, and people work hard to make it clear and also ensure that their results are significant.
And we have the software now that can quantify fluorescence across a geographic space.
And this paper is open access. It's in PLOS Pathogens, so you can follow along if you wish. pull over to the side of the road and open up your copy.
It's definitely worth my favorite experiment is this Lit A GFP experiment, just because the fact that it works so well in Figure 7 is really nice. Great. They must have been super excited. If we had their most exciting day in lab, I would guess that one.
That would be it, yeah.
To have a fusion to a protein that can then find its way to the cell wall is really cool.
Yeah, and just taking advantage of their knowledge of the biochemistry of the system to develop a marker for a particular event.
Mm-hmm. Exactly.
Thank you, Petra. I'm going to read just 2 short emails we have to close off the year. Heather writes, "Hi, TWiM team. I'm a lecturer and undergraduate advisor in the microbiology department at the University of Massachusetts Amherst. I'm teaching our major's Intro to Microbiology course this semester and wondering what memorable papers you have for young undergrads to read as an introduction to peer-reviewed data papers. I'm asking because I listen to TWiM when commuting. I have heard Michelle and Michael call out a particular paper as being a nice one for undergrads, but I'm never in a situation where I can take notes and then often forget once I hit my office and see my unread emails. But now I would love to find a really simple and elegant paper that is fun to review how scientists communicate findings to each other. I always promote Microbe TV podcasts among our undergraduates. I'm so thankful for your contributions. I am amazed at the time and devotion everyone puts towards this endeavor. Thank you, Heather." I thought, Petra, you would have a suggestion for
I think I really like reading these classic papers that go with the original kind of observations, because we teach students Avery, McLeod, and McCurdy, in our intro molecular biology class. But to actually read the paper is really actually wonderful. It is a little bit long for undergrads. I do it with upper level undergrads or with sometimes my grad students. But to read the actual paper behind, how do we know what we know beyond the textbook, I think is really helpful. I think actually one thing we could do maybe on the TWiM website is to, maybe on TWiM too, is to start posting papers we think are really good for undergrads, because I totally agree. We say them and I'm always not in a position to write them down when I'm listening to podcasts. Sure. But it would be nice to have that as a resource.
What do you think of today's paper that you did? Is that a good one?
I think it is okay. I think, again, the problem that I had clearly even talking about it is the names are very similar to each other.
Yeah.
I think it would be helpful. It would not be a good first paper, but I think it's a good paper if you could keep track of the names. Maybe I tried to color code them with different highlighters. That's one way to do it. I think the paper we did actually, and I'm going to use this in my own undergrad class next in January, is the paper we did on plasmids and the plasmids. That is actually an excellent paper, the one from from Hunter College.
Hunter, yeah.
I think that is an excellent paper. I'm happy to kind of go through and put papers up.
Add that to the show notes. I'd also like to add that a colleague, Dr. Nancy Borey, who is at Iowa State University and was recently recognized at the American Society for Microbiology with the 2020 Karski Award, also uses TWiM in her teaching for undergraduates. And she has designed a really nice adjunct to the TWiM podcasts that she's currently developing, and has been kind to engage us in those discussions. And so it's premature, but Dr. Nancy Borey is going to really make a big contribution in this area.
So Mark Martin on TWiM 180 also talked about a couple of papers he uses in his undergraduate teaching. So I'll put a link to that. And I will start a page of papers suitable for undergraduates, and we can add to that over time. That's easy enough to do.
And let's invite our listeners too, if they are teaching undergraduates and would like to recommend papers maybe that have already been done or that we should do for TWiM, and that could also then be used for
you could send those to us, TWiM, TWiM at microbe.tv. And one more letter from Marion. I just had a horrible thought and thought I would ask for your expert opinion. Would rising global temperatures put evolutionary pressure on fungi, select for higher temperature resistance? As far as I understand, our core body temp is our main adaptation against fungal infection. Maybe we should be thinking about an antifungal research program ahead of time just in case. for listening to Arturo Casadeval, this is what I understand is happening.
Rising temperatures are affecting infectious disease in lots of ways. For example, mosquitoes that we used to associate with countries that are closer to the equators, their range is expanding, and therefore populations that are vulnerable to those mosquito-borne infections are also expanding. And that's just one example.
Yeah.
Actually, Rachel Penchikowski in my department is studies evolution actually of a plant fungus, but she has found she's actually studying the change in its distribution and ecology based on climate change. And I think a lot of ecologists who study fungal pathogens in particular and are seeing changes that have to do with temperature and climate change. And I think, again, that we definitely should be looking at those, even though they're not human pathogens. They're certainly going to be reflective or paralleling what's happening with human pathogens.
All right, that'll do it for TWiM 279. You can find the show notes at microbe.tv/TWiM. Questions, comments, suggestions for papers, TWiM@microbe.tv. If you like our work, we'd love to have your financial support. Microbe.tv/contribute. We have a number of ways you can do that. Michelle Swanson's at the University of Michigan. Thanks, Michelle.
Thank you. Good to see you both. Happy New Year.
Petra Levin's at Washington University in St. Louis. Thanks, Petra.
Thank you, and Happy New Year to you, too.
Happy New Year to all of our listeners. Have a good one. Be safe, and have a great next year. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.